Hello everybody, let's start today's class. So, today we have been looking at these different structural aspects, right. We started from the primary structure, secondary structure, then we looked at these helical angles, axis and all. We looked at the different secondary structural elements, right. Last time we were looking at something known as motifs. Now, what we are doing, what we are going to do today is rather focus in this class is kind of take the next leap, go to another structural level which is referred to as the tertiary structure. So, what you will realize soon is as we go through the class is that once you have once we have the idea about these secondary structures, then essentially it is the way that these secondary structures pack, thereby giving rise to the tertiary structure. Okay. So, we will look at this tertiary structure in some details, right, as you will soon see. It actually gives an idea of how different secondary structural components are oriented with respect to each other in different proteins. Okay. Hopefully, we can realize that or be able to appreciate it a lot more after the class is over. So, when we talk about the protein's tertiary structure, then there are two things we are talking about. One is the packing. When we talk about the packing, what are the different types of packings? Well, it is very simple. The two major secondary structural elements are helix and beta. So, obviously, one would be helix packing, the other one would be beta sheet. So, sheet packing, and the other one would be, the third one is an obvious choice, is a mixture of these, right. It is the helix sheet packing. Now, what are the different structural classes? So, obviously, if you are going to have a helix packing, then it is going to be all alpha. So, that is what you have as all alpha, right, if you follow my arrow. Then if it is going to be a sheet packing, then it is going to be all beta, and if it is going to be a mixture, then it is going to be alpha and beta. So, in today's class, what we are going to talk about primarily are the helix packing giving to all alpha, or giving rise to all alpha rather, and the sheet packing giving rise to all beta. Now, the alpha and beta, I leave it up to you to read, and as I will tell you later, this alpha beta mixed is the most common structural element, and I want you to read this yourself in details. So, going forward, let us look at the alpha domain structures. So, why do we want to look at the alpha domain structures? Well, all of you know myoglobin. This is the first globular protein to have its structure determined. It is a predominantly alpha helical protein, hence the importance of alpha helical packing or alpha domain structures. So, these alpha domains are or alpha helical domains are frequently found as the trans membrane segments of membrane proteins. You must have seen this. Then structural proteins like keratin, collagen are composed of alpha helices. However, alpha helices are only marginally stable in solution. What I mean is that if you have, say, a protein having 8 or 10 alpha helices, and if I take one alpha helix out of it, all these alpha helices together, say 10 alpha helices together, make a stable protein, but the moment you take one alpha helix out of that arrangement or that packing, then that alpha helix in almost all cases or all probability would not be that stable, which means that alpha helices are marginally stable in isolation. So, hence you can understand that alpha helices have to be stabilized. So, how are they stabilized? If you read the first point, hence alpha helices are stabilized through packing of helices, the packing occurring via hydrophobic side chains. Now, obviously, if you are going to pack, if you are going to pack, then what is going to happen is you will be having a certain surface of the protein which is going to be turned away from water, which is your physiological solvent, right. In your physiological environment, water is in your physiological environment, and then you would be having hydrophilic residues which are on the outside. So, a coil-to-coil arrangement, as you can see at the bottom, the coil-coil arrangement of alpha helices maximize the aforesaid side chain interactions, right. So, this is an example of a typical coil-to-coil, and what you can see is if this distance is about 14 nanometer from one to the other, what you can see is here you can see the way these two different alpha helices are kind of wrapping around each other or coiling around each other. Okay. Now, going into this structural aspect in a little more detail, a very common aspect of this coil-coil structure is a repeat or is a sequence known as haptid repeats, right. Now, these repeats are very common in coil-to-coil alpha helical structures. Now, what I mean by haptid repeats, I will soon tell you, but first let us look at these amino acids. Okay. So, you see how many amino acids we have? We have A, B, C, D, E, F, G. So, you can understand So, that means A, B, C, D, D, E, F, G, these are 7 amino acids, hence comprising of the heptad, or this is what the heptad is made up of, specifically for the protein I am talking about. Now, if you look at the different amino acids, what you will soon see is, if you go from A to G, well, A, so if this NH2 is, if you follow my arrow, this is the N terminus, and then COOH, this is your C terminus. Now, this, as I was telling you, this is for a specific protein. Now, this heptad repeat is of leucine residue, sorry, this C should not be there, it is a typo, of this GCN4. It is a transcription growth factor, it is a coil peptide, helical coil, it is a transcription growth factor. Now, what you see is one of the most important things, as is colored in blue, the D position. The D position is occupied by leucine, which is a hydrophobic amino acid. Okay. Now, this has severe consequences and very interesting consequences, as you will see very soon, right. Also, if you go to if you look at the other amino acids like even E, you have your kind of share of charge or polar non-polar residues too. But then what is the use of this heptad repeat? Let us look at that. So, if you would remember this this coil which we looked at a couple of slides ago. Now, what you are doing is you are taking a cross section of it and then you are zooming into it. So, once you are zooming into it, what you see is you remember in the last slide we talked about this heptad amino acid, right, A to G. Okay. So, this is what you see, this is the red one is one helix, right, as one helix out here, the red helix, and the blue one obviously is the blue helix, and you are looking at this portion, right. So, this is how it looks like, it is kind of a bent portion. Okay. Now, moving forward, it says that there are 3.5 residues per turn. Now, if you talk about a helix though, if you remember our alpha helix, there were like 3.6 residues per turn. However, here we have a little bit less. Now, the reason being, if you kind of go back to that alpha helix discussion, the alpha helix was kind of a straight smooth cylinder, right. There were no kinks, we never talked about those in one alpha helix. But here you see the structure is not straight, both the alpha helices are to a little bit distorted, and hence this brings about a decrease in the number of residues per turn. So, there are 3.5 residues per turn. So, you can understand then the repetitive sequence of amino acids per 2 turns, 3.5 into 2 is called the heptad repeat. So, 3.5 into 2 makes it 7 amino acids, the heptad, and so every 7 amino acids you have kind of a sequence which is repeated. So, the amino acids in such heptad repeat are labeled as A to G, as I was telling you, and as I mentioned and also showed to you in the previous slide. In terms of the sequence, this A to G amino acids constitute what is known as the heptad repeat. Okay. And to kind of show it on these two helices, you can see out here. So, this is one, this this enclosed portion, this black line with the portion is enclosing, it is showing you the heptad for the red helix coil, and the other one is showing it for the blue one. Now, how are these packed? So, if you look at this, now you are before you were just looking at in the previous slide, you were just looking at the amino acids, right, A, B, C. You were not looking at the actual interactions. But now, if you think about it in terms of the helix, right, then what you see is as you go through, does not matter which way you go through, you go from here to here or from here to here down, you can see this D amino acids, which I said was leucine or hydrophobic, are typically packed against each other, which means, remember again a few slides ago we discussed that the helices cannot be stable in isolation. This means when they are stable, they have to be packed, and the packing has to stabilize this whole arrangement, right. So, in this case you can see there is one component which is hydrophobic, right. This is hydrophobic packing, because the D amino acid was essentially a hydrophobic amino acid, which is leucine. So, the D residue, as it says, is hydrophobic, it is either leucine or isoleucine. The one we are taking in this case is called leucine. Due to the heptad repeat, the D residues are seen to pack against each other in the coiled coil. Okay. This is because of the heptad repeat, that repetitive sequence of 7 amino acids. Now, this arrangement is often referred to as the leucine zipper. So, you know what zip is, you can zip it up like this, like zipping chains. So, you can see if you are going to zip down the helix coil, what you are going to look at essentially are leucine residues, and hence is referred to as the leucine zipper model or motif. Now, the residue A is also hydrophobic and forms the hydrophobic core. So, the residue A is also hydrophobic. Remember, we talked about this residue A to G. The A typically also comes out to be hydrophobic. Okay. So, what are some other interactions that are present now? It cannot be only hydrophobic interactions, it must be something more than that, because we have so many other amino acids. So, if you look at this, what we are what we are doing now is, if the helices we looked at, what we are you are looking at from the top view, that means these are the helices, right. So, so if I go back, so these are the helices, you are looking at the helices from the top. Okay, you are just looking at them from the top, there is a top section. So, because of the top section, you can see how these are arranged. Now, you see this green portion, you can see out here, the green portion is made up of these A and D residues, which are forming the hydrophobic core. However, you have also other amino acids which are not necessarily hydrophobic, but hydrophilic. So, then you will be having ionic interactions, which are essentially salt bridges between G and E, E and G of these two helices, and that is what you also see in the next panel, which is a side view. So, this one is the top view, and this one is you are looking at from the side. So, that is what it says, the E and G residues can form salt bridge interactions, as has been shown above, thereby further stabilizing the coiled coil structure. So, again, it pays to kind of look into this matter a little bit more, and what you have just realized is, once we looked into this matter, we see that how this isolated alpha helices, which were actually not stable in isolation, are being stabilized in presence of other helices. So, this is one type of packing we have. So, now when we talk about this packing, the leucine zipper packing, I mean, it is given to it is given a special name, it is called a knobs in holes model, because helices have to pack, and this is one type of packing. So, what I mean is, so this is one helix, helix 1, the red one, this is helix 2, the blue one. So, what you are seeing is, as it says, these are the projected positions of the side chains of the two helices. So, you are taking this, the red one and the blue one, and you are taking the side chains and putting them along the helical axis. Now, what you see out here is, please note, so here the inclination and here the inclination is different. So, you can understand, if these helices have to pack against each other, one of them will have to tilt or just move in an angular orientation, one of these helices with respect to the other. This is exactly what happens. If you go to the next slide, again you look at helix 1, you look at helix 2, and then you try to superimpose them. You cannot superimpose them just like this in this form, as it is shown in helix 1 and helix 2. You will have to do certain transformation, right. What do we see? So, this is what you see. You can see this blue one has been tilted a little bit with respect to the red one, which is helix 1, the angle being 18 degrees. Not only that, look at these, look at this D out here, if you follow my arrow, this D and this A, right. So, this D is surrounded by 4 other amino acids, and this A is surrounded by 4 other amino acids. Okay. To explain it a little simply, what happens is, on superposition after appropriate tilting, that means now you are tilting it, the side chains of the first helix. So, these are the side chains of the first helix, you can see these are the side chains of the first helix, they are known as knobs, right, as written in brackets, they appear between those of the second helix. So, these are the side chains of the second helix. So, what happens is, the moment you tilt it, you can see this F, G, A, B, the red ones which belong to the red helix, they are now as knobs appearing in vacant spots or holes between this, in the ones in the blue, P, A, G and F. Okay. So, this is important now. So, the way you have packed it, you just cannot pack it arbitrarily, you go to pack it so that you have vacant spaces and that you maximize the interactions. This is the way the interactions are maximized, and also you make use of the vacant spaces. That is why it is called the knobs in holes model. Holes means you have vacant spaces, knobs means these are the amino acids which from one helix chain, which can go inside the vacant spaces or holes of the adjacent or the other helix chain, giving rise to this coiled coil structure. Okay. So, the knobs in holes model, again, now if you try to zoom into that, remember we talked about this. So, if you go back, we talked about this D and A, right. So, if you now try to zoom into this, and C, then what we see is, so this is typical. So, this is your D, right. So, this is a D which is a leucine, and these are the respective amino acids from the other helix, right. That is why these two helices are so colored, differently colored. This is one helix, this is the other helix, these two together are forming the coil. So, as it says, leucines, which is the knobs of one helix, sit in the hydrophobic holes of the other helix. This is what you see. The D is leucine, D, A are primarily hydrophobic, but he is hydrophilic. Now, each side chain in the hydrophobic region of one of the helices can contact 4 side chains from the other helix. Now, this is exactly what you see out here. So, in a nutshell, this is the packing. Okay, this is the packing. This is the knobs in holes model that goes on in a typical leucine zipper motif. Okay, so this is again we are looking at the leucine zipper. This leucine zipper binds, so the GCN4 coil, it binds to DNA. So, this is the DNA. You can see this the septide repeat, and these are the leucines, right. These grayish spheres are the leucines, and there is one more way you can look at it. Again, here you can see this DNA out here, and these are the two coil helices with the leucines in between. Okay. So, this is typically what we have been looking at in the previous few slides. But the question is this the only type of arrangement we have when it comes to helix packing? So, let us look at it. So, another very common helix packing is this 4-helix bundle. So, you can see this here the red ones are your helices, and what you have done is, these are like the topology diagrams. You are representing helices as cylinders, and you have 4 helices forming a bundle. Okay. So, the red cylinders are the alpha helices, as I said. Now, the figure on the far right is a schematic view down the helix bundle. So, what you mean is, if you are looking at the helix like this, if you are looking at the helix like this, and this is the arrow you just saw, then again you are looking at, it is a kind of a top view, right. You are looking at, or the bottom view, whichever you look at it. So, what you see typically is, see these green residues out here, these green residues are packing in the core. Packing in the core means this is the core, this is the core, this is where the green residues are packing. That means they are giving rise to something known as a hydrophobic core. But what about the other residues? These red residues, these red residues are jutting out into the surrounding solvent, interacting with the solvent, surrounding solvent, and hence this is a hydrophilic. These are the ones which are helping, which are on the surface, they are helping to keep the total motif or the total 4-helix bundle protein solubilized in water. So, as it says, the green circles are the hydrophobic residues buried. So, this dotted line actually tells you the section of the hydrophobic residues, and the red circles are the hydrophilic side chains which are predominantly exposed to the surrounding solvent. Okay. So, this is the simplest and the most commonly occurring of the alpha helical domains, the 4-helix bundle it is. The relative orientations of the helices and the angles between the helices determine the folds. Now, this is important, how the helices will be oriented and at what angle will finally determine what fold this protein domain or this alpha helical protein domain takes. In most of the 4-helix bundle proteins, the helices are packed either according to the knobs and holes models, while in some the packing follows the ridges and grooves model. Now, the knobs and holes model I have looked at, yeah, we just discussed, but the ridges and grooves model, what is that? That is what we are going to come in discuss about next. Now, these are functionally very diverse, starting from RNA binding, electron transfer, so cytochrome based protein, and then polysaccharide cleavage and so on. So, this is this cytochrome B562, it is an electron transfer protein, as I was telling you. It is a typical 4-helix bundle. Okay. This is a a better picture of that. It shows, so not only that, this cytochrome, it is a heme protein, that means it has this heme moiety, if you see this heme moiety, and this is a histidine ring which actually is binding to the Fe iron out here. So, anyway, what it means is this 4-helix bundle of cytochrome harbors a heme moiety. Also, what you see is, if you look at this topological arrangement, so this N goes like this, from N it goes like this, then it comes like this, that means it is an antiparallel arrangement of your adjacent helices. Okay. So, antiparallel helical arrangement. So, some of the structural features, remember we wanted to talk about the ridges and grooves model, right, because we have already seen what the knobs and holes model is. So, but before going there, let us look at some of the structural features, so that we can get a better feeling of this. So, it is a cylindrical protein having a length of about 50 angstroms and a diameter of 20 angstroms. So, there is the length and then you have the diameter. So, the helices are longer than the average ones, they have about 17 residues per helix. The angle between the helices is in the range of 17 to 29 degrees. Now, this means, so this angle is essentially, we will again see later at what angle, that means at what orientation are these two helices with respect to each other. The adjacent helices are antiparallel, we saw it, and the helices do not bend significantly, but move apart to accommodate the heme moiety. So, they are not bending, but what essentially they are doing is they are just moving apart a little bit, so that the heme moiety can come in and be stationed out there. So, this is another protein, it is called the repressor of primer or the ROP protein. Now, its function is to bind to the RNA. So, it is a homodimer, it is a homodimer. So, homodimer means what you can see is, see this red one and the blue one. So, these two, so this the red one you can see there are two helices, one dimer, two helices, another dimer, and this is a homodimer, that means the same dimers, I mean the same sequence, they are just coming and packing against each other. Each polypeptide chain forms a helix turn helix. A helix turn is very simple, there is a helix, then you have a turn and goes to the next helix. Okay. Now, each subunit has a coil structure based on the knobs in holes model, right. That is how the helices are coiled, remember that is how the helices are coiled. And the two subunits now are packed against each other using the ridges into grooves model. So, what it means is, if you just go back one point, so these two helices or these two helices, right, these are the two dimers, they then come together and form the homodimer. So, what it is saying is that these two are packed against each other using the knobs and holes model, the other two are also packed against each other using the knobs and holes model. So, the two red helices knobs and holes, the two blue helices knobs and holes among themselves. However, when these are packed, that means when the red dimer and the blue dimer, they are packed against each other, what we get is the ridges and grooves model. That is the model they use to pack. That was with regards to this 4-helix bundle. Now, where does this ridges into grooves model really come into existence? It becomes really evident, right. So, before actually talking about the ridges into grooves model, let us look at this globin fold. This globin fold is a very common fold. Why? So, you talk about hemoglobin, myoglobin, these are the ones, these are the globins where this fold is typically found. So, what you see out here is, this is amongst the most important alpha structures, good. Then these are found in related proteins like myoglobin and hemoglobin, I just said, and there are 8 alpha helices A to H. So, you can see A, B, then if you follow C, then it is D, right, then E, F, right, F sorry, then G, then H. Okay. So, this is F, so this is F, then you can have you see a G out here, then you can see H out here. So, there are 8 alpha helices, and what is this space filling diagram? So, this is your heme moiety, and it is connected using via a proximal histidine, covalent linked proximal histidine to the iron atom, to the F helix. Okay. Now, what is different here as compared to the others? That means the ones you have just come across. You look at, especially first of all, you look at the orientations. You look at this helix B and look at the helix E. You see, these are not just like this, right? So, go back. So, these are not just like this. So, what they are almost orthogonal to each other. So, they are almost orthogonal to each other. If you can see, they are almost orthogonal to each other. So, if this is B, then E is coming like this. Okay. So, there are different arrangements. It is not a regular coil arrangement or the 4-helix bundle arrangement that you have just come across. So, what are the features of the globin fold? So, this would explain it a little more detail. The pairwise arrangement between alpha helices are different from those of the 4-helix bundles, that is what I just said. The sequential helices are not necessarily adjacent. Okay. So, if you go back, what it means is, you can see A, B, then C, then D, but E is more close to B than C, right. So, that means the sequential helices are not necessarily adjacent, or the way it looks is that H is very close to A rather than B being close to A. So, that means you will be having a sequence, the amino acid sequence, right, you number it, this has 153 amino acids, myoglobin has 153 amino acids, but not necessary that one helix and the next adjacent helix adjacent in the amino acid sequence would be very close to each other. Okay. Then the two helices at the end of the chain are antiparallel, which is G and H, forming a helix turn helix motif, but the remainder of the fold does not include any characterized or characteristic supersecondary structures. Except G and H, all packing interactions are between non-adjacent helices. Now, this is really important. Just being adjacent in amino acid sequence does not guarantee that they would be adjacent in packing too. So, this is how packing is different. This is how packing is different from the sequential arrangement of the amino acids. It is not necessary that adjacent amino acids or adjacent helices have to be packed against each other. It is absolutely not necessary, what you have just seen. So, then as I was talking about the packing of helices, see, in the packing of helices you will be having two things. One is, this is one helix, this is one helix, this is the angle between the two helical axes, and then the distance between the two helices. So, if you think about the different types of helical packing, right, then the one is the knobs in holes model. So, knobs in holes model, we have already seen this in the case of the Lewis zipper. Here this angle is 20 degrees. So, here you can see out here. So, this is the angle between the two helices, it is about 20 degrees. Now, for the ridges and grooves model, which appears in the Globin fold, this angle goes to about 50 degrees. So, considering alpha helix to be smooth, now remember when we started talking about this, that we never saw this when we were talking about a simple alpha helix, right. Just the secondary structure element, if you think about, if you go back and think about the alpha helix we talked about, we did not talk about these. But what we just said is, if based on the topology diagram, alpha helices can be considered as smooth cylinders, and hence we would expect that if you would have an alpha helix like this and another alpha helix like this side by side almost parallel, they would be maximizing the interactions, but that obviously is not the case. So, this is, please pay attention to this, this is where we start or try to see what this ridges into grooves model really means. Okay. So, this is a helix, if you look at the first figure. So, this is a helix, a polyalanine helix. The surface has the C beta residues, that the carbon atom in the beta position of the amino acids, right. So, what you can see is, these are the different amino acids, this is this one first amino acid, fourth amino acid, then 7th, 8th, 11th, 12th, like that, okay. And the ridges has been shown by lines, okay. So, please pay attention. So, for example, so if these are the surfaces where I am looking at the C beta, then look at this, these lines correspond to ridges. So, these 4, 8, 12 are amino acids and they form one ridge. So, it is like if you have seen mountains ridges, it is like one is up, then you have a group, then you have a ridge, then you have a groove. So, it is like ridge, groove, then ridge, a groove, and like that, okay. So, that is what you are saying, ridge, then you form a groove, ridge, form a groove, like that. So, that means here I am 4, 8, 12, these are the ridges. So, in between you will be having the groups, again you have 7, 11, 13, 15, 19, these are again ridge, in between you have a groove here, again you have 18, 22, right, these form again a ridge, and between this line and this line you have a groove, between this line and this line you have a groove. So, the lines essentially are your ridges, and between the two ridges you will be having grooves, right, it is like your valleys. Same here, if you look at the last figure, you can see there is 1, 4, 7, 8, 11, 12, 15, 18, and so on. Now, what is the difference though between these two? As I said, if you look at these amino acids, there is 4, 8, 12. So, they are spaced by 4 amino acids, like there is I plus 4. Same here, 7, 11, 15, 7 plus 4, 11, 11 plus 4, 15, 15 plus 4 is 19. The other hand, the last one, you can see these amino acids which are on the ridge, they are spaced out by 3 amino acids. Say for example, 1, 4, 7, spacing of 3, 8, 11, again a spacing of 3 amino acids, right. Now, this is important. What it tells you is this: if you take this, the second figure where we have 4 amino acids, that means there is a difference of 4 between the amino acids on the ridge. So, this is what we have. This is referred to as a plus minus 4n arrangement. So, plus minus means that if we consider 8, then minus 4 will give me 4, plus 4 would give me 12. So, this plus minus 4n arrangement, I plus 4, I plus 8, I minus 4 rows form the most prominent feature. That means, this is the most important feature or the most common feature in the ridges and grooves model. Then rows formed by residues 4, 8, you see 4, 8, 12 and 7, 11, 15, 19, they belong to this class as we just said. So, 4, 8, 12, 7, 11, 15 and 19. Then the C beta atoms in these rows are about 6.4 angstroms apart. So, these between these two the distance is about 6.4 angstroms, and from one residue to the next you got to have a rotation of about 40 degrees. Okay. Now, please try to put this in the context of the rotation we talked about for a screw axis or the screw symmetry that a helix possesses. Now, if you go into ridges and grooves model now, for the second case where now we have a spacing of 3 amino acids. So, this is what you get, this is plus minus 3n as it is I plus 3, I plus 6. The rows formed by the residues, what is this 1, 4, 7, then 8, 11, 12, 15, 18, 19, 22, they belong to this class. The C beta atoms in these rows are about 5.6 angstroms apart. So, you see they are closer than the previous one, and from one residue to the next we have a rotation of 60 degrees. That means, they have to rotate a little bit more, right. So, there are 3 amino acids, they have to be rotated a little bit more. Okay. Now, so the ridges formed by residues of one helix pack or intercalate into the grooves of a second helix, obviously, right. You will be having ridge of one, the ridge of another cannot fall on the ridge of another. So, it has to fall into the groove where you have the vacant space. If both helices intercalate through their 4 end ridges and grooves, then this angle is close to 50 degrees. Okay, remember this angle we talked about, this is the most common one. If one helix uses the 4 end groove and the other helix uses the 3 end groove, then it should not be a groove, it should be a ridge. There is a typo out here, this groove should be read as ridge. The resulting angle is close to 20 degrees. Okay. That means, this is the groove and this is the ridge. So, there is you cannot have packing between two grooves obviously. So, you can see that depending upon this 4n packing and 4n 3n packing, you give rise to different angles, one is 50 degrees, the other is 20 degrees. So, this is what I mean. So, this is the top row of figures, there is a 4n grooves and ridges. So, you can see here this angle is 25, here this angle is 25. Now, if you go to the last one, when we try to actually what happens is, if you try to take this and try to put this interface along with this helix, then you have to take it out of the paper by about 180 degrees, which is what you do out here, and then what you do is you flip it by an angle. So, here, so this angle out here is about this angle between these two, this this shaded one and this red one is about 50 degrees. Okay. This angle is about 50 degrees. However, if you go to the other one, which is the bottom one, here the 4 end grooves are the red and the 3 end ridges, remember I talked about the ridges. So, this is the groove, okay, and this is the ridges. So, what will happen now is, you can see the difference between this guy and this guy is, these two helices were oriented in the same direction, but these two were oriented in the opposite direction. Not only that, this is 25 and this angle is 45. So, you can see finally, when they are aligned, the angle between them is 20 degrees as opposed to this angle being 50 degrees. Okay. So, this means that depending upon your 4n 4n or 4n 3n, your orientation and hence the angle between these helices would be different. Okay. So, again coming back to the case of myoglobin, these are your cylinders, this one is a heme, this black one is a heme, right, this black line. You can see how this B and E are oriented with respect to each other. The same has been shown here. Okay. So, this is your helix B and this is your helix E, and you can see how they are almost having an orthogonal packing. This is in case of sperm whale myoglobin. Okay. So, this is what I wanted to tell you about alpha structures. Now, please keep in mind that there are other variations of this. There are other proteins which have many proteins which have different variations of these structures, but this is the principle that the packing is based on. Obviously, one is your knobs in holes model, right. and the other one is ridges into grooves model. That means one protein has the grooves, and the ridges of the, that means one helix has the grooves, and the ridges of the other helix come and fall into that groove. Okay. So, next talk would be beta structures. Here we have looked at all alpha helical structures. So, now what about the beta structures? So, let us look at this. So, this is also very interesting. These are the second most common class of protein domains. See, the second most common class of protein domains. What was the first one? The alpha beta, which I said you got to look up yourself. But I will be giving you questions though in the exam, please keep that in mind. Now, these are functionally the most diversely populated group. So, you see transport proteins, antibodies, enzymes and so on. The beta strands are arranged in a predominantly anti-parallel fashion. So, whatever we discuss, you will see it is an anti-parallel arrangement. They are classified by the topology. So, what I mean by that? So, what are the different classifications? One is up and down barrels. If you remember, for a beta sheet, we were actually using up and down arrows, right. So, that is what I mean. So, using those arrows, we can say what the topology is. One is the up and down barrels, the other one is your Greek keys, and the other one jelly roll barrels. Okay. So, these are the three which we are going to look at in some details in the subsequent slides. So, there we are considering the up and down barrels. So, this is the one which is having the simplest topology. Now, similar arrangement to Tim barrels, I have not discussed this, but without the helices and all strands are anti-parallel. Tim barrel, you can understand then, it says without the helices, that means the Tim barrels would be a mixture of helices, I mean alpha helix and beta. Adjacent strands are connected by short hairpin loops. We will take this one by one, and then strands adjacent in the amino acid sequence are also adjacent in the 3D structure. The interior of the barrel is hydrophobic and also has the binding pocket. What is going to bind there? We will come to it later. Okay. So, here what you see is, okay. Now, let us talk about this. Adjacent strands connected by short hairpin loops, right. So, you can look at these strands. So, suppose this is a N terminus, right, this is N terminus, this is C terminus where we are ending. Now, you can look at these, the beta strands they are being marked by these arrows. So, from when you go here, then you have this hairpin loop, it comes and connects to the next strand is connected to the next strand and so on until you come to the C terminus. That means, these adjacent strands are connected by these short hairpin loops, almost these hairpin loops, right. So, that is how the arrangement goes. Now, you can see the strands adjacent in the amino acid sequence are also adjacent in the 3D structure. You can see what happens. So, these are adjacent in the amino acid sequence and these are also adjacent in the 3D structure. So, they are also the hairpin loops, they are also adjacent close to each other in the 3D structure. That is how they pack. Now, because of this packing, now before I go there, if you take this and you look at this, so this is their topology diagram. So, the one you have at the bottom is a topology diagram. Here it does not actually show you the structure, it just shows you the connectivity using the arrows. So, this is what you see, right. This N you start, this is the adjacent one, okay, this hairpin loop, then arrow is in this direction, then the arrow goes in this direction. So, this is an anti-parallel arrangement and each and every adjacent beta strand is connected by this hairpin loop. Now, the way this is arranged, what will happen is, if you look at this in the middle, right, that means if you look at it in the middle, you can see the way it is arranged, it is kind of like this. So, that means there is a pocket inside and hence this interior is quite hydrophobic. So, this interior is quite hydrophobic and that is why it is called, it is hydrophobic and also has the binding pocket. So, binding what? It must be binding to something, right, functionally important possibly. So, that is what we are going to look at in the next slide. But before going to the next slide, just understand up and down barrels, what it means is, so see this is one is up, this one is down, this one is up, this one is down, this one is up, this one is down and so on. Essentially that is what it means, up and down, anti-parallel arrangement. Adjacent strands also adjacent to each other in the structure as they are in the amino acid sequence. A typical example of this is this protein, the retinol binding protein, RBP we call it. Okay. So, this is retinol. Okay. You see this protein, right, the retinol binding protein, you can see this 1, 2, 3, 4, 5, 6, 7 and 8, and then you see this is the C terminus, right. Now, what will happen is, in the middle you see this yellowish color and you have seen this chemical structure. This chemical structure is of that retinol. So, this is retinol. So, this retinol is bound in the interior hydrophobic pocket of this up and down beta barrel. That is why this you use this red circle portion, right. Okay. So, the retinol what it does is, I said that the interior is hydrophobic and it is a binding pocket. So, the retinol goes and binds there, okay. As the arrow shows, this is retinol. Now, the protein has 182 amino acids and it is a transporter of vitamin A, obviously, because retinol vitamin A, which is bound, which is getting bound to the hydrophobic pocket, it is being transported by this protein to tissues where it is needed from liver. Now, looking a little bit like zooming into the binding site a little bit more, you can see this is a retinol, right. See, in the previous slide, if you notice, the retinol ends with an OH group, the other ones are primarily hydrophobic. The same thing happens here. You can see, so these are the interior. So, you can these green ones are the different amino acids which line the interior hydrophobic core of your retinol binding protein. So, obviously, this is hydrophobic. So, it would prefer hydrophobic groups, right, hydrophobic atoms out there. Hence, the full almost the full retinol molecule is out there inside the hydrophobic core, except this OH which is polar, and hence it is coming out, getting exposed to a more polar environment. So, this means that it is a very good fit in terms of hydrophobicity, in terms of these interactions between the hydrophobic retinol and the hydrophobic amino acids which are lining your core. So, going forward, the hydrophobic part fits in a hydrophobic pocket. A hydrophobic part means hydrophobic part of retinol. Now, the binding site is lined with hydrophobic residues, as we are seeing. See, phenylalanine, 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 again phenylalanine, methionine, tyrosine. So, it is primarily hydrophobic, primarily by phenylalanine residues. Now, the hydroxyl group is exposed to the solvent, as I just said, right. So, that means that there is a very good arrangement or binding of this retinol inside the binding pocket, stabilized by the surrounding hydrophobic amino acids or interactions with these hydrophobic amino acids. So, what I mean is, then if you look at the alternating it should be alternating patterns amino acid sequence of retinol binding protein. What you see is out here. So, this is a strand number, this is a residue number, then this is the amino acid sequence 2, 3, 4, these are strands 2, 3 and 4. You are talking of the minus 41 to 48, 50 to 60, 71, 78. What you see is isoleucine, alanine, phenylalanine, valine, these are the hydrophobic groups. These are in between, you have these hydrophilic ones, glutamate, aspartate and so on. So, the hydrophobic amino acids are aligned such that these face the core. So, that means they are facing the core, the green colored with the red arrows, these are the hydrophobic amino acids. The polar charge and a few small hydrophobic are the ones which are exposed to the solvent. Okay. So, it is kind of alternate, one is hydrophobic, goes inside the core, the other one stays outside, the other one goes inside, the next one stays outside, that is why it is called alternating sequence. Okay. So, that was one about the retinol binding protein, and one thing you would have also noticed is that if when we are talking about this topology diagram, especially the one where we were just showing the connectivities, it looked a lot more simple. It looked a lot more simple than the actual one where you had all those strands coming in and you were trying to look at the structure. Then the next one is referred to as the Greek key barrel. Now, the Greek key motif is one motif which we had talked about before. Remember when we were talking about the supersecondary structures and all, the beta, alpha, beta and all, we had talked about the Greek key motif. Now, this is where you see it again. So, it is essentially beta. Now, what you see out here is one of the connections is not a hairpin connection. Remember, a hairpin connection is one where, so you take this and you take this, the two adjacent strands are connected by a hairpin loop, but you can only get a Greek key barrel where you do not have such a hairpin connection, right. That is why it says one of the connections is not a hairpin, your typical hairpin connection. So, then what it means is, if you look at this one, if you look at this topology and if you look at these two strands, this is n and this is n plus 3, that means you have a linker. You have a linker between n and n plus 3. This is not a short hairpin loop. Why? Because these are not two adjacent strands. One is n and the other one is n plus 3. And if you look at the, so this is the one which is normally observed, and if you look at the right one, so this is n and n minus 3. So, this is n and this is n minus 3. Okay. But both of these tell you the same thing. That means This is not a hairpin loop, because it is not happening, it is not linking to adjacent strands, it is linking two strands which are 3 strands, two strands which are 3 strands apart, like n minus 3 or n plus 3. But in this case, just to since we are already on this, to tell you, as it says, this is the one which is mostly observed, right. The n to n plus 3 is mostly observed, the n to n minus 3 is almost not observed. Okay. So, where is it found? It is found in a very important protein called gamma crystalline or crystallines, as a matter of fact. Now, gamma crystalline or crystalline, what it does is, see, crystallines, if you if you have heard about it, they are very important for eyes, right. So, for retina, for the transparency and everything, for us to see properly, because what happens is, you will see, you must have heard or you must have seen your relatives or maybe someone else, that when people get aged, they have problems with their eyes. So, they undergo this operation known as cataract operation, because what happens is, in that case, so they have a blurred vision, they cannot see properly. You know why they cannot see properly? What happens is, these crystallines, they start aggregating, okay, with age, some things not happen properly, the body does not function to its optimum. So, the proteins start aggregating, and the proteins start aggregate, so they become insoluble, and then they start precipitating out there itself. Once they precipitate out there itself, if they are not getting removed by anything, which they are not typically, then because they are precipitating, that means they are no longer soluble, they form an invisible screen for you or an opaque screen for you, and hence your vision gets blurred. So, this crystalline has this Greek key motif as a part of it, that means it is made up of this Greek key motif. So, it says it is found in lenses of your eyes, right. You can understand the importance of it. All of us have this. Each domain is built up from 2 Greek key motifs. Okay. So, this is one Greek key motif, this is the other Greek key motif. One connection is across the barrel between the 2 motifs. So, there is one connection, because if you have these 2 motifs, you have to have one connection which connects these 2 motifs. So, you can see there is a connection from here to this here. So, this is one Greek key motif, this is another Greek key motif, and you can see, right, and you can see this is the connection. So, this is domain 1. So, this is domain 1, this is domain 2, the right hand side is domain 2. Again, this is the topology diagram. So, what you can see in the topology diagram is, let us see what I have in the next slide. Well, what you can see in the topology diagram is, I will show that to you is, so just look at this. Here you can see this 8 beta strand, this 8 beta strand is linked to one strand in domain 2. So, this is the linker we are talking about. So, again, gamma crystalline is made up of 2 Greek key motifs. One is forming domain 1 and the other one is forming domain 2. What we will do in the next slide is, we will look at one of these domains in some detail, at least the connectivity. So, you can understand, you can appreciate this topology, why this topology diagram is so important, why people talk about this topology all the time when you talk about protein structure or structural hierarchy and all. Okay. So, moving on, let us look at this one domain. This is one domain, remember. Okay. So, I am just taking one domain, does not matter which one I take, whether this one or this one, I am just taking one domain, right. So, now look at this. What I have is, you can look at the strands, you have 1, strand 2, strand 3, then strand 4, then you have strand 5, 6, 7, 8. See how different this is from what? The up and down barrel. What did we say in the up and down barrel? We said in the up and down barrel was that each strands, or the strands which are adjacent in the amino acid sequence, are also adjacent in the structure. Here it is not so. Here it is 3, there is 4. Adjacent to 3 we have 8, okay. Now 7, adjacent to 7 obviously we have 6, but then 5, 6, 7, 7 and 8, these are far apart, aren't they? So, the beta strands are arranged in 2 Greek key motifs, right. 2 Greek key motifs, this is 2 Greek key motifs, you can see this is one Greek key motif and the green one is the other Greek key motif. So, that is what it says. It is found by strands 1 to 4 colored in red. So, remember we are looking at only one domain, 2 Greek key motifs, one domain, this is one domain, it is colored red, it is made up of strands 1 to 4, then the other domain from by the green ones, strands 5 to 8. Now, let us think about the topology, right. How do we figure out the topology? 2 ways you have to figure out the topology. One is, you got to figure out what are the adjacent strands. Once you have, so that is what you focus on first. Figure out the adjacent strands, start drawing the arrows. once you have the adjacent strands, then start putting in the connectivities. Let us see whether we can have it here. So, suppose say I start with 8. Okay. So, listen, this is 8. If this is 8, which is a part of the green one, then what is the one adjacent to 8? Obviously, this is 3. You can see out here. Sorry, let's go forward. You can see this 3, hence 3 is adjacent to 8. Good. Now, obviously 8 is the last one. So, this is the C terminus one. Okay. Now, this is 3. Now, adjacent to 3, what do we have? We have 2. See, this is where we have 2, right. See, this is where we have 2, and this 3 is adjacent to 2. So, this is the loop which is connecting 2 to 3. Now, adjacent to obviously is 1. So, this is 1, the 1 is N terminus. So, if you go from 1, so this is 2, the 3 goes out there, right. Then obviously this is 4. So, if this is 1, adjacent to this is 2, I have written 2 here, adjacent to 8 is again 4, I have written 4 here, right. Then 4, what do we have adjacent to 4? We have adjacent to 4, we have 7. So, see, 4 is kind of in between 1 and 7, that is what we have, 4 between 1 and 7, right. Then if you have 7, then what we have is this is 7, then this is 6, right. So, this is 6, 6 and 5, you have this hairpin loop, 6 and 5 you have this hairpin loop. Then look at this, 3 and 7 we have this connectivity. Remember, we are talking about n and n plus 3. So, essentially this I am sorry, let us go back on that, sorry. So, this is 8, this is 7, you can see this is 7, this is 8, that is the connectivity you are looking at. Okay. Then this is 5, this is 6, this is 5, this is 6, right. What else? Then we have 4, we have 3, that is we have already shown. So, here 4 you can see is connected to 3, then this 3 is a internal connector to, that is what we have. So, as I said, if you start putting in the adjacent strands and start putting in the connectivities, you can see how simple this thing becomes. You start the N terminus with the strand 1 and then you end at the C terminus with strand 8, but again adjacent strands are not necessarily the ones where the amino acids are also adjacent in sequence. Okay. So, this is how a topology diagram kind of helps you to realize what the actual structure is without the connectivities or without the actual orientations. Again, please keep in mind that here the arrangement is anti-parallel in nature. The last one we are going to talk about in terms of this tertiary structure are the jelly roll barrels. So, the jelly roll barrels, they are it is a polypeptide chain is wrapped around a barrel core like a jelly roll. So, you can see if this is this whitish one is a barrel, and you can see it is kind of wrapping around like a jelly roll. It is found in a lot of receptors and enzymes. Now, here also 2 Greek key motifs in the jelly roll barrel. So, you have 2 Greek key motifs. So, this is one motif. So, this is the other motif. So, this is the topology diagram of the jelly roll structure. Okay. Now, I will come to this again. So, but just realize out here what happens is it is kind of very similar to what we just did before for that Greek key motif. You see 1, 2 and then you have 3 out here, right. Then there is 4, okay, 5, 6, then is 7 and there is 8, right. Again they are not adjacent, that means the ones which are adjacent in sequence are not necessarily adjacent to each other, right. Now, there are some very interesting things out here. If you look at this one, and kind of zoom into it, spend a little more time on it, see what you have, right. Okay. Remember, if you look at this, there are 2 wrappings. So, this one wraps on top, this one also wraps on top, then this one also comes down, there is one at the bottom too. Now, look at this, do we have that in this topology diagram? So, you see 7 to 8, this runs across the barrel, this is on top, again 4 to 3, 3 to 4, this runs across the barrel, this is on the top. So, you can see this is what we have, this is 8, this is 7, this is running across the barrel, just transversely across the barrel. Then the next one was 3, so this is what we have 3 and this is 4. You can see these are the top ones, as I said, these are the top ones which are running across the barrel. Then we also have 2 which are down. So, these are the 2 ones which are looking at. So, this one, if you follow my arrow, it is this one and also this one is between 2 and 3 and this one is between 7 and 6. So, look at 7 and 6. So, this is 7 and this is 6, you can see this bottom one and the next one is between 2 and 3. So, it is very simple. So, you understand you have just taken this, you can start from any any strand, you say you can start from 2, you can then after that you can put in this arrows up and down depending upon this anti-parallel arrangement. After you have done this anti-parallel arrangement, you just put in the connectivities and it becomes so simple. A seemingly complicated structure, when you look at the topology diagram, it just looks a lot more simple without all the connectivities, without all the space filling models, without the orientations, the minors and everything. So, you see in the bottom we have 2, as we said, top we have 2, and then we have these hairpins here, here and here. Okay. So, that is we have I have told to you or talked to you about topology before, but not to this extent. Here you can understand that in this better sheet packing, this topology is really becoming handy, isn't it? That means using the topology you can you are being able to really simplify the overall 3-dimensional structure. Okay. Please try this yourself. I have shown this to you, but I guess what you should do is you should take this only and then try to figure out how I can connect these. So, if you compare compare all these beta barrels, it is B, it should be beta, B should be read as beta. So, you can see this is the up and down, right. This is the gamma crystalline like, and this is the jelly roll like. So, you can see in the in the gamma crystalline, this is where you have these the top 2. In the jelly roll, you can see this is the top 2, and then you also have a bottom 2. In this case, you have one bottom also out here. So, this kind of brings us to the end of this class and to the end of the discussion of the tertiary structure. So, now what I need you to do is, because I have already spent a lot of time on this, is to look at these alpha beta mixed domains, look up these alpha beta mixed domains by yourself, and then also look up coordinate structure. So, coordinate structure is you guys all know this. I mean, I do not have to go into that much details, right, but I can just tell you that if you have different domains like different tertiary structures, like consider hemoglobin, each hemoglobin is a tetramer. So, each monomer is having its own tertiary structure, and when these different monomers they combine, they form this tetramer. This is a coordinate structure. So, essentially multi-domain proteins, right, where they have different domains and all these they pack, giving rise to this coordinate structure. Okay. So, I guess that is what I will end. Please do look up these structures, these are very fascinating. You take up any look up this protein data bank, take up these beta structures or alpha structures or the mixed, the one you have to look up. Try to see whether you can relate their orientations, especially for helix packing, with what we discussed, the knobs into holes or the knobs in holes or the races into grooves. And for beta sheet, if you are looking at those structures, then you think about the topology. That is why the topology makes it so simple. That is why people are talking about the topology all the time. Okay. So, we will meet in next class. Thanks.